So, to begin with, we can talk about the different types of blood vessels that there are in the body, or the composition of the different layers of our blood vessels. So, for their general structure, all of our blood vessels, all of our larger blood vessels, our artery and veins, have three layers. An inner, what's called tunica intima. So the first layer is the inner layer, called the tunica. It has two names, either interna or intima. And this is a simple squamous epithelial layer. So that when you get down to the capillaries and when you go from larger vessels to smaller vessels, the outer layers are going to start getting smaller and smaller, so that by the time you get to the capillaries, you just have a tunica intima. This is the smallest possible layer across which we can have the diffusion of our respiratory gases and our nutrients. So that's our first layer. A specialization in veins is that the tunica intima has valves. And that's because our veins aren't really under blood pressure. So in order for blood not to pool or not to backflow, there has to be valves that catches it. And so we'll talk about the blood pressure that's acting on the blood in veins in a little bit, but just be aware that in veins, the tunica intima contains valves. In arteries, it does not. Arteries are under blood pressure from the heart, but veins are not. So all of the blood pressure from the heart gets dispersed in the capillary bed. So this layer is going to be selectively permeable to small solutes like our respiratory gases, glucose, and things like that. Some other things about this layer is that it can actually secrete chemicals that help with vasoconstriction or dilation. So it can secrete vasoconstrictors or vasodilators. Under normal circumstances, this layer repels blood cells so that they don't stick to it. But if there's damage, then this layer will start secreting chemicals that will attract platelets to the area. So we'll say normally repels blood cells so that they just flow right past it. But with damage, secretes chemicals that are going to attract platelets to the area. If we're having an inflammatory response, which we'll go over in great detail when we go over the immune system, they secrete chemicals called cell adhesion molecules, or they express what are called cell adhesion molecules, and those are going to allow the leukocytes to stick to them and roll along the surface of the tunica intima so that they can slow down and squeeze out to the tissue. So during the inflammatory response, they express cell adhesion molecules, also called CAMs. Cell adhesion molecules, which are called CAMs, or abbreviated CAMs. And these will allow our leukocytes to kind of roll along the surface. So if you look at the tunica intima, it's this simple squamous layer of cells, and if there's damage or during the inflammatory response, they'll stick up these cell adhesion molecules off their surface. And what that does is it allows our white blood cells that are coming by to stick to it and roll along the surface of our blood vessel. When we get to the inflammatory response, when we talk about the immune system, we'll talk about this more. But then these cells can slow down and actually squeeze between the cells to get out to the damaged tissue. So that's going to be another important part of what the tunica intima does. It's going to secrete these, or express these cell adhesion molecules that will slow our leukocytes down. So I'm going to erase this top part while you guys write the functions. So our deepest layer is the tunica intima. Superficial to that, not the most superficial, but superficial to the tunica intima is our next layer called the tunica media. And this is made of smooth muscle cells or smooth muscle tissue. In general, this is the largest layer of most of our blood vessels. It's got smooth muscle cells, it's got some collagen, and sometimes some elastic connective tissue, and our elastic arteries. It's got elastic connective tissue. And the thickness of this is going to vary depending on what type of vessel you are. We'll talk about the specific types of vessels in just a minute. But the production of this is to produce what's called, or the function of this is to produce vasomotion, which is the motion of our vasculature. So it can either dilate or constrict depending on what's going on in the body. So that's its big function, is to produce vasomotion. Which is dilation or constriction. So the vasomotor center is going to be able to trigger vasoconstriction or dilation depending on the needs of the system. Are we good on all our functions of the tunica intima? All right, so the most superficial layer of our blood vessels is called the tunica externa. That is often continuous with the tunica externa of neighboring vessels. So a lot of times you'll see arteries and veins that are running together have tunica externas that are continuous with each other. And it's just kind of helping to give some support to the structures. And the whole point of this is to anchor our blood vessels and our nerves and blood vessels that are serving the blood vessels enter through here. Those blood vessels are called vasa vasorum. So the function is to anchor our blood vessels. And the next function is to allow for the entry of the vasa vasorum, which are blood vessels that are serving the blood vessels. Largest blood vessels, it's thought that the inner layers of the tunica media is being served by the blood flowing through it, but then the outer layers of the tunica media has to be served by these vasa vasorum. Any questions on the general composition of our blood vessels? We're going to talk about specifics in just a minute. Okay. So as far as our different types of blood vessels go, we can start with arteries. Arteries are what are known as our resistance vessels, and that's because they're resisting blood pressure. So these are our resistance vessels. And I actually want to make sure that I don't, okay, here we go. So arteries are our resistance vessels. And the first type of arteries that are exiting our heart are called elastic arteries. These are also called conducting arteries because they're conducting blood away from the heart. We call them elastic arteries because they have a high proportion of elastic connective tissue, which allows them to resist blood pressure. All that blood pressure, when the ventricles contract, they need to be able to expand, and then they recoil when the ventricles relax, and that is going to allow us to maintain blood pressure even though the ventricles are relaxing. So they contain elastic connective tissue, which gives them elasticity, which is the ability to stretch and recoil. If all these vessels did was stretch, then when the blood, or when our ventricles relaxed, you'd lose blood pressure and you'd faint or something. So by having the ability to recoil, then they can still exert pressure on that blood even though our ventricles are relaxing. So these are going to be the largest of our arteries, and they have thicker walls than all of the rest of our arteries, and they've got thicker walls than the veins that are going to ride with them. So they've got a pretty thick tunica media, and they are going to be conducting blood away from the heart. So we also call them conducting arteries. The next type of arteries are called muscular arteries. Muscular arteries are also called distributing arteries, and they're distributing our blood to specific body regions. So these are also called distributing. Because they distribute our blood to specific body regions. Muscular because their tunica media is their thickest layer relative to the other layers. So, and it's thicker relative to the whole artery, our whole muscular artery, than is the tunica media even in our elastic artery. So it's got a very thick tunica media. So they're going to branch away from our conducting arteries and distribute blood to specific body regions. The wall is about 2/3 smooth muscle, if you look at it. So it's 2/3 smooth muscle. So it makes them very muscular relative to every other type of blood vessel. So if you think about like where blood is going, our elastic arteries are like highways, and our muscular arteries are like exits off the highways that are taking you to a specific body region. And then our last type of artery is kind of like the street that'll get you to exactly where you're going. So if we're good on this, talk about the next kind of arteries, which are called arterioles. These are the smallest arteries, and they are going to be pretty small. They only have 1 to 3 layers of smooth muscle and very little tunica externa. So these are our smallest arteries. They've got 1 to 3 layers of smooth muscle. And little to no tunica externa. So these arterioles contain what are called precapillary sphincters, and they're these little muscles that are going to be able to constrict to stop blood from going into a capillary bed or dilate to allow perfusion of a capillary bed. So we have a lot more capillaries in our body than we could fill with blood all at once. We don't have enough blood to fill every capillary bed. So capillary beds will constrict their precapillary sphincters if that area of tissue doesn't need oxygen and nutrients right now, and then ones that do need our nutrients and oxygen will relax those precapillary sphincters. So these contain precapillary sphincters that will relax to allow perfusion of a capillary bed, or that will dilate to pass the capillary bed by. So arterioles are the smallest of our arteries, They contain 1 to 3 layers of smooth muscle. The bigger arterioles have 3 layers. As you get smaller and smaller and are approaching a capillary bed, you'll get down to 1 layer. They have very little tunica externa. And then they contain these things, the precapillary sphincters. You should remember sphincters are orbicularis muscles that are going to relax to allow perfusion of blood to the capillary bed, or they constrict to prevent flow and you just bypass that capillary bed. All right, from our arterioles then, we'll pass into a capillary bed. Our capillaries are our exchange vessels. So this is where we're going to have nutrient and gas exchange. So this is how we're going to get our oxygen to our tissues or in the lungs, get our CO2 back out to the lungs so we can breathe it off. These are very, very small, and we've got 3 different types. The first type is the most common type, continuous capillaries, and our continuous capillaries are going to be present in most of our tissues, and we just have 1 layer of endothelial cells that are held together by tight junctions, but contain what are called intracellular clefts. So these are found in most tissues. And these are going to be endothelial cells. So simple squamous epithelial cells, but it's like the endothelium of the blood vessel. And they're held together by tight junctions, but contain intracellular clefts. Intercellular clefts that we can have the movement of materials. So continuous capillaries are going to be most of our capillaries, and they're going to be where we're having our nutrient and gas exchange. Found them in most tissues of the body. They've got these simple squamous epithelial cells that we call endothelial cells because our endothelium is I guess another name for the tunica intima. It's the innermost layer, and it's epithelial cells. They're held together by tight junctions, but they have intercellular clefts that's going to allow for a movement of material. Okay, our next type of capillaries are called fenestrated. Fenestra is the German word for windows. So they have lots of little holes in them. So they contain fenestra, which are like holes that allow for the passage of larger solutes. You find fenestrated capillaries in areas where we need to have filtration. So places like your kidneys, where we're filtering our blood to make urine. So in our liver we have a different kind because it's doing a bunch of different types of filtration. So we'll say this is in tissues that require filtration. So an example would be the kidneys. We are filtering our blood to produce urine. So our fenestrated capillaries are going to allow most of our larger solutes, other than like big proteins. So big proteins shouldn't be able to squeeze through there, but things like waste molecules can pass through our fenestra. Okay, then our last type of capillaries are sinusoidal capillaries. Your book just calls them sinusoid capillaries. A lot of books will put an AL, sinusoidal capillaries. And our sinusoidal capillaries have large open spaces. So they're like irregular blood-filled spaces, and this is going to allow for the passage of really large materials. So we find these in the liver, and the spleen, and the bone marrow. So these are like large, irregular, blood-filled spaces. That allow for the filtration of really large particles, like dead blood cells. The spleen, where we're filtering out dead red blood cells. You find them in the liver. And the liver is going to be important to have these big spaces because it's producing a bunch of plasma proteins that have to get out. So you find them in the liver, the spleen, and the bone marrow. Bone marrow, because we've got to get whole new cells out. So anywhere we've got to do really large particles, you'll find sinusoidal capillaries. Our next type of vessel called a venule. Our venules are our smallest veins. And actually I should say our veins are what are called capacitance vessels. And that's because they've got a high capacity to hold blood. So they act as a blood reservoir, and at any given time they've got a lot of our blood flowing through it. So 55 to 60% of your blood is held in veins. So they have a high capacity to hold blood. Our smallest veins are called venules, and they conduct blood away from a capillary bed. Our venules are going to converge to become our small and medium-sized veins. So our next veins are our small and medium veins. And our smallest is when our venules converge to form our small veins. And then our small veins converge to form bigger veins, our medium veins. And our small and medium veins are going to be companion vessels with our muscular arteries. So these are companions to muscular arteries. And in our small and medium veins, this is where we start seeing our valves. So your venules don't have valves, but your small, the larger of your small veins do. So this is where our valves appear. The valves in the tunica intima appear.
Really quick, so the venules, they converge to form the small, and then
The small converge to form,
medium.
yep, medium. And the point of these valves is to prevent the backflow of blood. And again, that's because our venous blood is not under blood pressure. All that blood pressure gets dispersed when we hit the capillary bed. So we'll talk about the different pressures acting on the system in just a minute. Then our large veins are companions to our elastic arteries. Definitely still contain valves. These These guys are running into our elastic artery and returning blood to the heart. All right, so we call our veins blood reservoirs because at any given moment they have most of the blood in the system. So if we look at where the blood is found throughout, or if we have 100% of our blood and look at where it is at any given moment, about 18% of it is going through the pulmonary circuit. The heart has about 12% of it. The whole systemic circulation has the other 70%. Our systemic arteries are carrying about 10%, and 55% of it is hanging out in our veins. That means that our veins are holding most of our blood at any given moment. That's what makes them the capacitant vessels. They are blood reservoirs because they hold most of our blood at any given moment. So when we talked about ways that you could alter stroke volume, we said that one way to do that was to increase venous return. So how you can do that is you can pump more blood out of those veins. It'll come out of that reservoir and enter the heart so that we have increased stroke volume, which would help to increase cardiac output. Any questions on that? Does it make sense how our veins are our blood reservoirs? They're holding most of our blood. We can access that if we need to for increased activity in things. And we'll talk about how we move blood out of veins in just a minute. Any questions so far? All right, then let's talk about the pathways that blood vessels follow. So a simple pathway is the most common circulatory pathway. And what happens in a simple pathway is that we have an artery leading into a capillary bed. And the capillary bed leads, or I should say, an arteriole. Leading into a capillary bed. Our capillary bed will lead into a venule. Our venules will converge to veins and bring it back to the heart. We'll get through our arteries and so forth. So most of our pathways are simple pathways. But there are alternative pathways. So we've got what are called anastomoses and portal systems. And anastomosis, there are different types of anastomoses, but an anastomosis is when, we'll say, multiple blood vessels are coming together. So, multiple blood vessels merge. So, in an arterial anastomoses, you would have many arterioles kind of coming together and forming like a complex network, and then emptying into a capillary bed, and then being drained just by one venule. A venous anastomoses is the opposite, where you've got one little arteriole leading into your capillary bed, and then the capillary bed is going to drain into many venules. These venules can be another complex network. Eventually they'll come back together and dump into a vein, but if you've got many venules coming together, we call that a venous anastomoses. And then an arterial venous anastomoses is what's called a shunt, and this is when you can bypass your capillary bed altogether. So we would have, I'll put our capillary in the background first. So we have a capillary bed in the background, but we can shut those precapillary sphincters, and the arteriole then will dump right into a venule. So we can bypass the capillary bed altogether before returning to the general circulation. we call it a shunt, an arteriovenous anastomosis, arteriovenous anastomosis coming together, or a shunt. Then the last type of ulterior or alternative pathway that's possible is a portal system. We've already met one portal system, the hypothalamo-hypophyseal portal system. We'll meet another one when we get to the digestive system, the hepatic portal system. And what happens then in a portal system is that we have blood coming in through an arteriole. It will pool through a capillary bed. The capillary bed, that first capillary bed, will drain into a portal vein that brings the blood to a second capillary bed, and then it dumps into a venule. So in a portal system, the blood passes through 2 capillary beds before joining the general circulation. Questions on pathways of blood. So this is the most common. Simple pathways are the most common, where an artery or an arteriole is going to dump into a capillary bed. We'll have nutrient and gas exchange. Then that waste-filled, CO2-filled blood will enter a venule and return to the heart. We've got different types of anastomoses that are possible, and then portal systems as well, but those are pretty uncommon. All right. So capillary beds then are going to be this area where we've got a lot of small capillaries, and we've got an arteriole leading into that, and a venule leading away from that. So our arteriole, on the arterial end, we have these precapillary sphincters, so that can close off the capillaries at the arterial end. And if we close them off so that they are all constricted, so we'll say on this side we're constricting, then the blood is going to go through what's called a thoroughfare channel and dump into the venule. So, thoroughfare, I don't think I'm spelling that right. Through our thoroughfare channel and pass right into the venule. So not all of our capillaries can be perfused with blood at any given moment. So we have the tissues that are in greatest need of oxygen and waste, or oxygen and nutrients, will have their precapillary sphincters open. If you just got oxygen and nutrients on the last pass through, then you can constrict these precapillary sphincters and pass straight through to the venule. If we've got a capillary bed and these tissues are nutrient and oxygen low, then that will allow the precapillary sphincters to dilate, and the blood will be able to perfuse the capillary bed so that we have nutrient and gas exchange. So on this side, we'll say our precapillary sphincters are relaxed, and that figure in your book, I think there's a figure in your book 20.5 that shows this in greater detail than this little picture I'm drawing. But we've got our precapillary sphincters relaxed, and then this blood will be able to enter the capillaries, and we'll have nutrient and gas exchange. We call this perfusion of a capillary bed. When the blood is able to flow through it, we'll have nutrient and gas exchange. We'll drop off CO2, drop off nutrients, drop off hormones, all that stuff, And then on the venule side, we'll pick up our CO2, pick up our waste, and that blood will exit into a venule.
Thank you.
Good on this? All right, so then what are we going to exchange in capillary exchange? We've got different types of capillary exchange I'll go over in just a minute, but things that are leaving our capillaries are going to be things like oxygen, glucose, hormones, what else is going to leave? Water. The tissues need water. All of those things will exit the capillary beds. Things that will be picked up or enter the capillary beds are going to be things like CO2, wastes. We can actually pick up hormones from the glands or excess hormones. So for hormones, we'll say from glands or excess. Oh, this marker at this side of the board is awful. We can pick up minerals from bones in the capillaries. So if parathyroid hormone and calcitriol are in the system, we can break down our bones and get those minerals out into capillaries and pick them up and bring them into the system. And then we can also pick up water. Wait. So water would be another thing that can enter a capillary bed. And we're going to be able to have all of these things moved by 3 different mechanisms of capillary exchange. The first is diffusion, and diffusion is always from movement of a material from an area of high concentration to low concentration. Diffusion in the case of capillary exchange is the most important because this is how oxygen and glucose get out. So this is the movement of materials from an area of high concentration to low concentration. As far as our tissues are concerned, oxygen and glucose are the most important things that tissues need. Those are both moving by diffusion, so that makes diffusion the most important mechanism of capillary exchange. So O2 and glucose are going to get to the tissues this way, and then CO2 is actually going to enter the blood from the tissues this way also. So they're all small enough to use diffusion. And so CO2 will leave the tissues and enter the blood by diffusion as well. Good on diffusion? Okay, the next mechanism is called transcytosis. We break this word down. Trans means across. And then cyto is the cell. And then osis is our movement. So we're moving things across the cell. So there are going to be 3 mechanisms involved in transcytosis. And the first one is pinocytosis. This is also called cell sipping or cell drinking. And that is going to be, if we look at our endothelial cell, then when pinocytosis is happening, we get these small little vesicles that are bringing in amounts of extracellular fluid. So things that would be in that extracellular fluid could be like ions or something. And so pinocytosis is the cell sipping, and we get these little vesicles that are bringing in extracellular fluid. And whatever's in that extracellular, whatever small solutes are in that extracellular fluid. So this would be things like ions, minerals, things that are too big to move by diffusion, but are small enough to be just kind of sipped into the cell in these small little vesicles. So the cells sip something in, they've got to be able to get out on the other side. As far as capillary exchange goes, I think I've got phagocytosis next, but that's not going to happen in our capillaries. So I'll cross it out. What is going to happen is we're going to have to be able to get this stuff we just sipped in to the other side. Oh, okay, so pinocytosis is a type of endocytosis where we're bringing in a vesicle, but pinocytosis is good enough. And then exocytosis now is how we're going to get this, what we just brought in. It's going to go across the cell through the cytoplasm and be dumped out on the other side. So this would be dumping materials out of cell in a vesicle. So if you're a little endothelial cell and you just use pinocytosis to bring something in, it can travel across the cell and be dumped out in an endocytotic vesicle so that it can get to the other side. We are not going to be moving a whole lot of stuff this way. It's just small amounts of extracellular fluid that we're taking in. So how we're going to get lots of stuff to move is our next mechanism, which is called bulk flow.
So this transcytosis, is that the same thing as vesicular transport?
Yes, because we're using vesicles to transport things. It is in the book. It's one of our mechanisms of capillary exchange.
779.
.780's got our next process, bulk flow. Oh, where's my bulk flow? Before we talk about bulk flow, it's actually good to talk about the different pressures. So bulk flow is going to be how we get lots of stuff to move, but we've got different pressures that are going to cause it. So hydrostatic pressure is the pressure that a fluid exerts on a membrane, very generally. And in the case of our blood, the pressure that is being exerted on the fluid is blood pressure. The pressure from our ventricles pushing on blood. So it's coming from the ventricles, and it's the pressure that the blood exerts now on our blood vessels. The next type of pressure that's moving through a capillary bed is what we call colloid osmotic pressure. And osmotic pressure is always the pressure on water to move from a hypotonic solution to a hypertonic solution. So we'll look at how this applies to our blood in just a minute, but for osmotic pressure, it's the pressure on water to move from hypotonic to hypertonic. Hypertonic being the solution that has a bunch of solutes in it. Hypotonic has more solvent than solutes. Colloid osmotic pressure is the osmotic pressure that exists in the blood due to all of the stuff basically that's in the blood, so that when we look on the venule side, we'll see that there's a lot of pressure for water to move back into the vessels, because relative to the outside, with all the plasmas that, all of the plasma proteins that don't get filtered out and all the blood cells that don't get filtered out, it's really hypertonic. So our colloid osmotic pressure is the pressure in blood due to solutes, which are blood cells and plasma proteins primarily. So this figure is figure 20.9 in your book on page 780. We'll talk about bulk flow in a minute, but all of those pressures are what is acting on things in the system. Then the last pressure is the net filtration pressure, which is the difference between our hydrostatic pressure and the colloid osmotic pressure. So filtration is the movement of substances then out of the blood, and so the pressure for that to happen is going to be due to the difference of this. So this is the pressure to cause filtration. We'll talk specifically about what filtration is in just a minute, but we can calculate this by taking our hydrostatic pressure and subtracting our colloid osmotic pressure. And that will give us our net filtration pressure, and that will tell us which way solutes are going to move. So these different pressures are going to allow for the last type of capillary exchange, which is bulk flow. Bulk flow is the movement of large amounts of fluids and whatever solutes are dissolved with it in one direction down a pressure gradient. So there are two different types of bulk flow, filtration and reabsorption. Filtration is the type of bulk flow that typically happens at the arterial bed of or at the arterial end of a capillary bed. And it's due to the hydrostatic pressure on blood. So you have much higher hydrostatic pressure on the arterial side than you do colloid osmotic pressure. So you have net filtration pressure to move out. So filtration is the movement of particles out of the blood on the capillary end due to hydrostatic pressure, which is our blood pressure. So we could say it's the movement of things out of blood on the arterial side of the capillary bed, because our hydrostatic pressure is greater than our colloid osmotic pressure. So the blood that's coming into our capillary bed is full of water, it's full of nutrients, it's full of our respiratory gases, it's got lots of stuff in it. So as it's coming through there and we push that fluid through those capillaries, we're going to squeeze it all out, because the hydrostatic pressure, or the pressure that's pushing the fluid out, is greater than the pressure of water to move back in on the arterial side. That's what causes filtration. The next type of bulk flow that we have is reabsorption. And reabsorption is the movement of fluid out of the tissues and back into the blood, because colloid osmotic pressure on the venule side is higher than hydrostatic pressure. So we'll have a net movement of fluid back into the blood. So reabsorption occurs at the venule side of the capillary bed, and it's the movement of fluid back into the blood, because our colloid osmotic pressure is greater than our hydrostatic pressure. So it's the movement of fluid from the tissues back into the blood. Because colloid osmotic pressure is greater than hydrostatic pressure. This colloid osmotic pressure isn't enough to return 100% of the things that were filtered out back into the blood. So about 15% of the fluid remains, and is going to be picked up by lymphatic vessels that we'll talk about for the next test. So for reabsorption, we'll say this Reabsorption reclaims only about 85% of the fluid that was filtered out.